Looking forward.
Thank you. It's a great pleasure to be here today. I want to introduce CRISPR-CLEAR, that is a new CRISPR screen modality to investigate the relationship between genotype to phenotype nucleotide level resolution. So as we know, last year, we finally have the fully assembled genome thanks to long-read sequencing. And as Eric Lander said, back in 2000, we got the book. It's hard to read. I think it's still hard to read. And the problem is that there are regions outside the coding space that are really hard to interpret. But still, they cover the majority of the genomic space. And more importantly, most of the disease-associated variants are in this space. So it's really important to study the function of these regions. And we know, thanks to several studies, that key controlling elements in this space are enhancers that can fine-tune gene expression in a cell-type specific way. And we have different ways to potentially discover them using GWAS, EQTL studies, chromatin profiling, and as well trying to understand their target genes using chromatin contact assays. However, if we really want to understand their function, we need to potentially perturb them to assess causality of these sequences. So we are lucky to have amazing technologies based on this CRISPR system. And if you're not familiar, this system is composed by a Cas9 protein, and then there is this guide RNA, this short RNA that can be essentially easily synthesized. And the system can be programmed to modify almost any part of the genome, assuming that you have a short sequence called PAM. And what the system will do, will find the target site, introduce double-strand breaks, and the cell will try to repair, introducing small insertion deletion. And over the year, people have evolved this system, and now we have CRISPR-I and CRISPR-A that you can use to epigenetically modify, for example, the genome to repress or activate region of around 200 to 1 kb. And more recently, we have these amazing enzymes like base editors and prime editing that allows you for base editing to create specific substitution in the order of 1 to 4 base pair. More recently, we have also prime editing that use reverse transcriptase to rewrite really precisely short part of the genome. So we have a really nice toolbox to play this game. So we can play essentially battleship against the genome and try to find the critical spots. There are different ways to play this game. And today I want to talk about one way to play this game that is called CRISPR tiling screens. So CRISPR tiling screens, now that you know what are these editors, you can essentially tile as many perturbations as possible in a region of interest. For example, with epigenetic perturbation, you can discover de novo enhancer elements because the resolution is quite low. And then with genetic perturbation, like CRISPR-Cas9 base editor, you can try to dissect this element and try to find subregions that often correspond to transcription factor binding sites. So just to enter a little bit more in the detail, after you design your perturbation, what you do, you essentially infect your cell with a lentiviral transduction, this small, rectangles color your perturbation. And the idea is that you want to have, on average, 1 perturbation per cell. And then here, after you wait a little bit, these edits are happening. And then what you can do, you can try to study a phenotype of interest. For example, can be the expression of a gene nearby your enhancer, can be a cell surface protein, anything that you can sort. And what you do by sorting, you divide your cells into populations. And now, given that you lentivirally integrate these sequences, you can sequence the 2 populations, and you can essentially study the enrichment of the perturbation in the 2 populations. And given that this guide RNA map to the genome, you get a nice 1D signal that may point out to the important part of the region that you want to study. A few years ago, we developed the convolution framework called CRISPR-Serve that, starting from this count, create this nice map that point to the critical part of the enhancer, and with some statistical assessment. And this was the work of Jonathan Su, a student who was in my lab. So, but this was in 2018, it was like a few years ago. So in my lab, we tried in collaboration with the Bauer and Serrugia lab to improve these screens. And today I want to present this new modality that we call CRISPR-CLEAR. And the key idea is that we want to increase the resolution. We want to go past a small region to actually find the critical nucleotides in this regulatory element. So the first step is the same. So you design your perturbation, you infect the cell, one perturbation per cell, you divide the cell into population, and just understand the screen, just the recap, the classic way that we call count-based approach. Again, you sequence the guide RNA integrated in your cells, and you count them. But the problem with this is that you actually don't see, you don't observe the edits that you're making to your regions. So that's the first limitation. The second thing is that this guide RNA have variable efficiency. They are not uniform. So this can confound the analysis. And if you put these two things together, you have quite low resolution, because what you measure is this guide RNA enrichment. And what you really want to do is like trying to measure the functional edits. So we reason, why don't we change this framework and just sequence the region? And that's essentially the revised version of the screen, in which we actually perturb this region. But now this time we design primers to sequence this region. So, and after you sequence, you get alleles. And you have now a read count of alleles. But now, like from the allele, maybe there is a way to recover the nucleotide importance. And if you have a nucleotide important, then you can start to think about transcription factor binding modulation of this perturbation. So to go from the allele to the nucleotide importance, we collaborated with Martin Jankowiak, that was at the Broad Institute, and that this really nice Bayesian variable selection method called millipid. And really high level, what we do, like we encode the allele with a binary encoding, and then this is your X matrix of the genotype. Y is like the allele score that is kind of the allele enrichment. And what we want to do with this method, given this genotype, this is like a proxy for your phenotype, you want to recover this beta that best explain this relationship. And given that it's a Bayesian method, you also get this posterior inclusion probability, that is the probability that this particular nucleotide score is important given your data. And also another thing of this framework that I really like is that depending on how much data you have, we shrink your beta towards zero if you have low evidence based on the data that you have. So now we have all the ingredients for this screen that we call CRISPR-CLEAR, and I want to show you the first demonstration of this. So for this, we focus on these NAL6 cells that are a model for leukemia, and then we're trying to study this CD19 gene that is a cell surface marker for B cell, and we wanted to study this region here that seems to be an enhancer. There is some conservation, this is a chromatin accessible site, there is also some ketone acetylation. So what we did was to delete the entire region. And as you can see here, you can see that this is the normal, the wild type. If you delete one copy or two copies, you see a nice reduction. So we believe this is an enhancer for this gene. And then we designed our screen. So these are the details. So we use, again, the CD19 enhancer. This is the region we are modifying. The phenotype is the fluorescence of CD19 because it's a cell surface marker, so we can use an antibody. And then we design around 200 guide RNA, and we have two base editors. One that is a C to T editor, cytosine, and the other one is an adenine base editor that is A to G edits. One is this NGG PAM, that means that you can target only if you have NGG. The other one is this SPRY enzyme that is amazing. You can essentially tile almost all the region. So the first thing that we did was to check first, based on the flow cytometry, if this antibody is good, then what you expect to see is a shift. If you delete the entire enhancer, you want to see the cell reducing this fluorescence. So we have a good dynamic range, and then we have like some individual guide, based on positive and negative control that just assess the dynamic range of individual guides. And this is the full library. That means that these are the cells where we don't recover, we don't see the individual guides, but we see that some cells respond to that. So there are like some informative guides. So now what we did was, we took these two populations, we extract DNA, and we have like alleles and allele and guide RNA, and we run the two methods, so you can see how much more you can learn with this new version. So this is the output of CRISPR-Serf. So this track here is the log change for this EvoCDA, and this is like for the log change for the other base editor. This is the deconvolution. And if you zoom in, you kind of see that there are like some regions that are, maybe slightly enriched, but there's not much power, to be honest, in the screen. And, nothing is statistically significant if you correct. There is this region that, is interesting. There are like two transcription factors that we know that are strongly associated, to this cell type. But, this screen clearly doesn't have the resolution that we want. So if you look, if you look the output of CRISPR-CLEAR, you get instead this beautiful high-resolution map that highlights the importance of each nucleotide. So let me zoom in, because it's really hard to see there. So this map looks like this, in which this is the reference sequence. And then, like the color nucleotide here correspond to the sites that you can edit. And there's like a weird way, this is rendered. So you should see that some of this would be gray, but you can infer from this. Essentially, base editing cannot modify everything. So in this map, if you see like a color or a square, means that you have some modification, but and then you have this beta that is recovered from the method that tells you like how much this substitution is disrupting CD19 fluorescence. And here you have the two screens, and you see that we can modify different nucleotides. And then the size of this square is like the peep that essentially is telling you if it's 1, you are really confident that this nucleotide is important. If it's close to a dot, essentially, we don't really trust it. The nice thing you can do here is that if you combine the two, actually this is rendered correctly. So you see that this is the reference, the gray are the things that we cannot modify. And here I put together the two screens, and you start to see that you have a lot of information about several nucleotides. You can create also this kind of disruption logo that tells you like for each nucleotide, how likely is that this nucleotide will change, CD19 fluorescence. And then, below you can put motif log, and now you can start to play this game comparing, the canonical motif logo with the disruptive sites. This is for Icarus, and you can see clearly that, the modification that we make essentially are not compatible, and as expected. In PAX5, there is a nice thing actually that I like. So here you see this A to G edit is quite, important, and PAX5, doesn't tolerate this G, but this A to G edit here is also like we can edit efficiently here, but there's nothing here. And if you look in the PAX5, actually we're helping the binding site. So that's why, to me, this is a nice, convincing example. And based on this, then we validated the two guides that can create these alleles, one for the EvoCDA and one for APE. And in both cases, this is the negative control and positive control. In both cases, we see a nice reduction. So I believe this is a really nice way to go from a large region up to nucleotide resolution and really learn this basic principle. So in summary, we have this new CRISPR modality. We're really excited. This is the first demonstration that we have. And there are like the key 3 take-home messages is that with this screen, we can perturb and measure edits at endogenous loci. We can recover functional nucleotide at higher resolution, and we can prioritize transcription factor binding site. But in addition, we can also assess their nucleotide preferences. So this was a really large teamwork between my lab, Daniel Bauer lab, and Davide Serrugia lab. So they generated all the data that I show you today. And, like I want to thank people in my lab, in particular Bashir that is leading this project, as well as Zain and Justin, Daniel Boerting and Marlena, and also Sandra and Anna in Davide Labs also to provide experimental data. And Martin that was at the Broad really helped, with this Bayesian framework, and also my funding sources. And I will stop here with the questions. Thank you.
Great talk, and I hope that soon we will use this method to identify new enhancers in genome. So you are using about 200 guides for tiling using in this method, right? So what is your priority? It's location tiling, or it's also you incorporate off-target effect of some of these tiling guides because they can be everywhere, right? And they can do something that you hope that it's effect, but in reality it's not there because of some location or multiple repeat elements, things like this.
Yeah, so the way I think about this, like if try to understand, I'm just trying to repeat to make sure I understand. So you're talking about, like, in here you can tile really large regions, but here we tile a small one. So you're talking about
there is some guides that can be this huge off-target effect.
Oh, off-target effect, yes. So for off-target effects, you can do in two ways. In my lab, we have some tools that potentially can predict off-targets. They are never perfect, but you can mitigate this effect. And the other thing that you can think about, like if you really tile densely, even if you have an off-target, you have nearby guides. So if you have like consistent signal, right? So then you can, envision that, like, yeah, maybe there is an off-target. And also, if you're really careful about this, you can actually test. There are like experimental assays to capture that. But yeah, this is a good question. For this, we just did something simple, try to avoid things that had less than 2 mismatches, against the reference genome.
Thanks. I have a question here.
Okay.
Same kind of question about off targets. So
yes.
The first question. So if you only look at 2 mismatches, depending on where these mismatches are, you can actually still have off targets. You might also have parallel genes. You
yes.
might have the same region and so on. So how do you actually know the effects you see on those off targets?
Yes, that's a good question. I mean, if you're really careful, again, you can run off-target assay. You can, you can enumerate the off-target in the genome. You can do amplicon sequencing and see if you have edits. And also, you have to think about this off-target somehow should change something, maybe in trans, and then it changes also the CD19 expression. So depending on the phenotype, this can be quite confounding. In this case, you have to think that this off-target somehow needs to change the CD19 expression, right? So, but you can be more or less careful depending on your goal. And I think that there are like ways actually to mitigate this. So you can, sacrifice maybe some guides.
I know
right?
there are ways. I'm just curious what you did.
I did, I did here like up to 2 mismatches, but most likely, is what we see here, this shift. You have to think about whatever off target we see should change expression CD19 specifically. So you have to envision a mechanism for this off target to modulate CD19. That's why in this case we didn't bring the big guns against off targets. Because this is probably a first demonstration. But we have tools in my lab. We have a tool called CRISPR-Me that we're planning to use for other screens where we can enumerate up to 7 mismatching, also RNA and DNA bulges. So we are capable of doing that.
I have another question.
Yes.
So you said that 98% of the genome is non-coding, 95% of all the SNPs are in those regions. How feasible is it that we do this for all the cell types we care about? So how far away are we from really understanding what's going on? If you would use a technology like yours.
So actually, this project, I listened to this slide, IGVF, is a large initiative where instead of targeting an answer, we're targeting variants. And in that case, you don't need the tiling. You don't need to have 200 guys for one element, but you can design guys against SNPs. And actually, there is a talk from my student Jayang I will present on Thursday. I will show some ideas about this different screening modality. It's a really good question.
So looking forward to those talks. So thank you very much.